Okay, so I'm going to be talking today about some of that more basic research that Alan touched on in his introduction, sort of looking at the underlying neurobiology of Fragile X syndrome, and specifically longitudinal development in Fragile X. Okay, not quite sure how to advance. Yeah, I've tried all of those. Maybe the clicker? Oh, something worked. Okay, so I aim the clicker at that.
Yeah, no, it's radio. Just
oh, okay. All right. Got it. Okay, great. So, as I know, I'm sure we're all familiar, Fragile X is associated with greatly reduced FMRP, which results in neurological and cognitive deficits, as well as social deficits that overlap with the characteristics of autism spectrum disorders. There's been a lot of research, in fact, I'm sure you'll hear about some of this today, research that is looking to develop targeted treatments for Fragile X syndrome. What I'm going to talk about in my talk is how the underlying neurobiology can help us really design the most effective targeted treatments. So neuroimaging is able to help us design effective treatments because we can really target the neurobiological systems that are affected in Fragile X instead of or in conjunction with targeting behavioral systems or symptoms. Neuroimaging can also help us identify objective metrics of treatment efficacy. So we can actually look at neuroimaging biomarkers, so the volume of a region, for example, in conjunction with or instead of behavioral or cognitive functioning metrics. And then finally, longitudinal studies are able to help us identify particular developmental windows when specific treatments may be maximally effective. So the study I will be describing today is a longitudinal investigation, a large investigation of boys from ages 1 through 13. And I'll be talking about our investigation of brain development, behavior development, and some brain behavior relationships. So longitudinal brain development is what I'll start with. And first, I'm going to show you here this lovely, a bit messy, but also I think very lovely graph of brain volume changes in the bilateral caudate. So you can see the caudate volume is on the y-axis and age is on the x-axis. So we're tracking caudate volume from age 1 through 13/14. And the thin lines represent individual data points. So that's one individual's growth trajectory. And the thick lines represent group-wise estimates of growth trajectories. So the first thing you might notice, well, first I'll also point out that the Fragile X group is colored in green, and the red group is our autism spectrum disorders group. So this is individuals with idiopathic autism spectrum disorders, that we're sort of using as a comparison, sort of a symptom match to the Fragile X syndrome group. So the first thing you'll notice is a great separation of all the green and all the red lines, which is quite unique for a neuroimaging result like this. And that is because the caudate is well known to be greatly enlarged in Fragile X syndrome. And so this is not a new finding for Fragile X enlarged caudate. However, what's unique about this graph and these results is that we can see that in the same individuals, caudate is in fact enlarged across these years, ages 1 through 13. And next I'm going to highlight a few other regions showing longitudinal growth curves. So the bilateral hippocampus and the amygdala, which are both involved in, important for the study of Fragile X syndrome. The hippocampus, of course, is critical for learning and memory. It's a region that's known to be enriched in FMRP very early on in development. We can see the matched symptom group, this time, first of all, we can see quite a divergence in the trajectory, so the shape of the two lines. Again, the Fragile X group is in green and the matched symptom comparison group is in red. We can see a roughly linear, steady growth in the comparison group versus a much more curved and delayed growth in the Fragile X group, but they do start to catch up. Starting around age 7, they have some rapid increase in growth. And the red asterisks indicate that ages 3, 5, 7, and 9 are ages in which there's a significant difference in volume. There is no significant difference in volume at age 11 through 13. So again, quite different growth trajectories here for the hippocampus. For the amygdala, as we know, is a key player in the social brain. You can see these trajectories look roughly more similar and are much flatter than the previous one I showed you. So here, there's actually no significant differences in volume across these ages measured, and there also is no significant difference in trajectory. Now, what's interesting is that the groups do seem to diverge, let's see, starting around age 9, and I think it will be interesting to follow these individuals further out in time to see if this trend does, in fact, continue. Although both, let's see, and it's also interesting that both groups do start to increase around age 7, significant increase in amygdala volume. Now switching gears, I'm going to talk a bit about longitudinal behavior development, so the other side of things here. So here we're looking at the same kind of growth trajectories, but this time on the y-axis is the ADOS, as we know, the sort of gold standard for measuring autism symptoms. And again, it's from ages 1 through 13. Fragile X is in green. First, you might notice much more overlap in the green and red lines, which is, from my experience anyway, much more typical of looking at behavior. It's a little more fuzzy sometimes than the brain imaging. But we can see that around the toddler age, there seems to be quite a large difference between the two groups, whereas later on, around 10 through 13, the groups seem to look more similar. And also we notice the Fragile X syndrome group is increasing in autism symptoms. So this measure is actually, if you score worse on this measure, that's indicative of higher levels of and more severe autism symptoms. So higher score, worse outcome. So as you can see, Fragile X starts off somewhat better than the ASD group, but they end up increasing in autism symptoms over time. This might suggest that early interventions could be particularly important for individuals with Fragile X syndrome at that early toddler age before they, even though at that age they may not be meeting clinical thresholds for autism, as we can see, there is likely to be an increase in symptoms over time. And now I want to talk about putting the brain with the behavior. So looking at early brain structure measured early in development. Can we use that to predict later outcome? The answer is yes. So I looked at early brain structure, the volume of the caudate measured at age 1 through 3, as a predictor of later ADOS scores, ADOS score being measured at ages 10 through 14. So this is a single time point within the age 10 through 14, but that was the age of the participants. And it turns out that caudate volume accounts for roughly 33% of the variance in later ADOS scores. And this is significant after accounting for total brain volume, suggesting that caudate volume may be a reliable, useful, specific biomarker for later outcome. And this relationship was only found for the Fragile X group. So when we look at a matched symptom comparison group that has the same or even perhaps more autism symptoms, as you saw in the last graph, this relationship does not hold. Now, of course, the caudate is just one region of the brain. We have to still look at various other regions and perhaps methods that can include variation of the whole brain as a potential biomarker, but this is an important first step, and that's that. So just to briefly summarize, the caudate is enlarged early in life and remains so through age 13. Behavior growth trajectories suggested that early interventions are very important in Fragile X syndrome, especially for later behavioral outcome. Early brain structure significantly predicts later behavior outcome, and early brain measures could be useful biomarkers. And finally, I just want to underscore that these types of results are really only possible with a longitudinal design. So following the same individuals throughout development is really critical to understanding these neurobiological differences and then in turn informing treatments that are perhaps going to target neurobiology. And of course, I'd like to thank everyone who worked on this project. So much of the work was done at Stanford. We're also working in collaboration with the University of North Carolina and also El Camino Hospital. This is one of our star participants who is indicating with his thumbs up that he enjoyed his experience visiting Stanford. And I think we have time for questions if anyone would like to ask one.
That's our propaganda slide, right?
Or propaganda, yes.
I was
Yes.
just wondering, looking at that, is there an effect of medications? Are these kids on different medications at different times? And I'm sure that the numbers aren't enough to actually say anything
Yeah.
definitive, but is there any sense that you have that perhaps medications are influencing some of these outcomes, either in terms of anatomy or behavior?
That's a great question. So the question was about medications influencing outcomes. And as you alluded, the numbers are quite small. What I can say is that most of these kids are on medications of some sort, and that is an area that we do want to look in. My inclination is that, yes, of course, medication is affecting brain growth and behavior, and we'll have to see how that pans out. What I would point out for the, let's see, let me go back to the behavior development growth curves. What I can say with a little bit more certainty is that this autism group, I can hypothesize why they're showing a decrease in autism symptoms over time. So this is a unique group that was brought to us very early in their development, ages 1 through 3. So they were most likely very brought to the attention of clinicians and researchers and clinicians, and were targeted for early interventions. So they likely had more intensive behavioral early interventions, and that is likely to have caused that decrease. So if we can apply the same thing to Fragile X, could be quite useful in conjunction with, of course that would not replace any kind of pharmacological target, but it could be useful on its own or in conjunction with a pharmacological.
Well, and I might add too, from the last connection where Jen makes the early brain connection to behavior, at that young age, very few children were on medication. So, in terms of the impact of medications at that age and predicting behavior, that might be a cleaner kind of look at the brain data at that point.
Yeah, I would imagine that would be a future confound when you're following a lot of people for a long time.
Yeah, and just the range of medications that are being used, right now, and if you do try to break them into kind of even categories of medications, it still becomes,
Yes.
So I have a question from the chat from George Hinkle. Is a high ADOS score indicative of someone on a higher behavioral problem?
That is correct. A higher score on this measure indicates more or more severe problems. So basically, this is a social behavioral assessment where an experimenter engages in play-type activities with a child and basically provides opportunities for social interaction. A score of 0 means totally perfectly normal social interaction. Most people would probably score, a 1 or a 2 because we're not all totally perfect. But basically, you're getting a point for every abnormal social thing that you either do or fail to do.
And also, does a large caudate indicate a higher ADOS score?
Well, I'm going to argue yes, it does in the Fragile X syndrome group. So this is what my analysis using regression showed, that a larger caudate volume at age 1 to 3, yes, was indicative of a higher ADOS score, which indicates a worse outcome. And if you remember the graph from a couple slides back, the higher Fragile X as a whole is associated with having higher caudate volumes.
So, this is actually working. All right, so I was just whispering to see if I could get it to respond. So I think, Amy, you were saying that if you go to the graph of any of them, like with the spaghetti plots on them, our little guys, they're usually almost never on medication, right? If anything, maybe they're on stimulants,
maybe, but
a handful.
maybe by like 5 we started, but
Well, so now that's really interesting. Following up on Mike's comment, can you go back to the hippocampus trajectory slide? So, as you point out, Jen, the hippocampus is of interest for a variety of reasons to Fragile X, just from a basic brain behavioral standpoint, from the standpoint of gene expression, FMR1 gene expression, early in development. But the other reason I've always thought the hippocampus is really interesting in Fragile X is because it's one of the regions of the brain, certainly not the only one, that is vulnerable to stress-induced insult, so to speak. And, you know, in some ways, you might think about Fragile X syndrome, particularly as kids get into school-age years, maybe even before, and certainly after, as peri-traumatic stress disorder. It's not post, because there's that tendency for hyperarousal that really starts to amplify around school-age years and into adolescence, where the stress response, as measured by behavior, by psychophysiology, by cortisol, they're all indicative that individuals with Fragile X respond to stress, but they overshoot and they have a much more difficult time reestablishing a stress response, post-stress response homeostasis. So now looking at that, getting to the point, the hippocampus, one of the things that has been proposed to be actually a mechanism of antidepressant response to serotonin, selective serotonin reuptake inhibitors, is the generation of neurogenesis in the hippocampus. It's very interesting. And so, you know, following up on Mike's point, we should probably look at how many of our kids get put on SSRIs that might account for some of that change in hippocampal morphology, because that could be pretty interesting. I actually have wondered for a long time about this balance between the tendency for overgrowth in the brain overall in Fragile X syndrome, most of it neuropile, but and the fact that the hippocampus is smaller, and wondered if, in fact, the hippocampi in individuals with Fragile X are being insulted by chronic stress and their physiology that doesn't allow them to adapt appropriately. And so when I see something like this, it really makes me wonder about all those different pieces thrown in. The stress response, possible insult to the hippocampus, SSRIs, neurogenesis, and so this is something we should really think about. I don't have any great insights today other than to say we really need to think about this.
Absolutely. Yeah, agreed. And the other thing that I would just point out when looking at these graphs is that there is no curve on here for typical development. So we don't know if, are they catching up to, as the Fragile X syndrome group is increasing, but are they catching up to what's typical, or are they still quite delayed there?
Well, we do have those data.
It's not on this graph.
It's not. I was going to say yes. we have the data, so we could do that.
The data is, yes, to be added. Any other questions?